Hi Bio 101, this is Dr. Lennon, your friendly neighborhood biology professor, with a primer on how enzymes function to help you prepare for lab. We've talked about enzymes in previous lectures, so we'll just do a quick review. Enzymes speed up a cell's chemical reactions by lowering energy barriers. We're going to talk a little bit about what an energy barrier is. But first, let's review what an enzyme does. A catalyst is anything that speeds up a chemical reaction within a cell, or speeds up a chemical reaction in general. An enzyme is a type of catalyst that is a protein. So our enzymes are proteins, which means they are going to behave in the way that we discussed how proteins behave in Chapter 3. So flash back and think about that for a moment. We know enzymes are important in speeding up chemical reactions within cells because, as I mentioned in an earlier lecture, without them, it's highly unlikely that your cells would survive long enough for the chemical reaction to take place. So they can speed up chemical reactions at least 1 billion times. The enzyme that you're going to be working with in lab this week is catalase. So I encourage you to refer to your lab handouts and take notes as I go through this quick introduction or even sometimes review of enzymes. Remember, when we're talking about chemical reactions, this is going to involve the making and breaking of bonds. All those chemical bonds that we talked about in earlier chapters, that's what we are referring to now. What the enzyme does is it makes the chemical reaction more likely to proceed. Now sometimes I think about this like a matchmaking website. You go to the matchmaking website, you give it your information, it searches its database, and it comes up with some possible partners for you. Someone for you to maybe go out on a date with, someone with whom you might have something in common. You could form a bond, like a chemical bond. That would speed things up. Your other option would be to just go out there and meet a lot of people, talk to them, go out on lots and lots of dates, and eventually you may find someone with whom you would bond. So an enzyme is kind of like that. It's a matchmaker. The idea is you would eventually find that person, but it may take longer and it would take a larger investment of energy. So an enzyme reduces the amount of energy and time it takes for a chemical reaction to take place. The energy that we talk about with enzymes is called activation energy, or the energy of activation. And it's usually noted as EA, energy of activation. This is the initial energy that's needed to start a chemical reaction. It's that initial push of energy that you need to get a chemical reaction going. Another analogy that I like to use is it's also like a down payment. So let's say you want to buy a house or a car or something else really expensive, but you need a down payment in order to buy that item. What an enzyme does is it reduces that payment, that initial payment that you need to purchase that item. So that activation energy is kind of like a down payment. And regardless, whether we're talking about matchmaking, whether we're talking about large purchases, or we're talking about chemical reactions, reducing the amount of energy you need initially allows that reaction, that purchase, or that match to be made more efficiently. So an enzyme catalyst lowers the activation energy, it takes less energy to get the reaction going, which means it's more likely to happen. It happens more quickly, it happens more easily, and it actually happens. Because you can imagine that if you just couldn't save up enough for that down payment, you'd never actually get to buy that item. Here's kind of a cute illustration of what we're talking about. So what we're looking at, on the left, we have kind of two graphs here. We'll think about them as graphs. We have the amount of energy in the system. And on the bottom, it's telling you with and without the enzyme. So this one is without the enzyme. We have our reactants, and then we have this activation energy barrier. So you can see this is kind of a large barrier. We need to push those reactants up this amount of energy, put that amount of energy into the system in order to get a reaction to take place. So this is what it would look like without an enzyme. This is what it would look like with an enzyme. And of course it's going to vary depending on your type of enzyme. So this is just a cute, literally, example. So in this example, this is our activation energy barrier, and it's greatly reduced by our enzyme. So it's much more likely that that reaction will be able to proceed forward to get our products. When you look in your textbook, or you look in pretty much any of the literature about this kind of work, this is really the kind of thing that you'll see. And we'll come back to this in Chapter 5. So if we look at this graph, we see free energy on the y-axis, the amount of free energy, and the progress of the reaction on the x-axis. The black line is showing us the course of the reaction without the enzyme. So we've got our reactants at this level of energy, but we need to add this much energy in order to get our reaction going. So without the enzyme, this is the example of how much energy we would need. Once we get to this point, the reaction will proceed and we'll get our products. With our enzyme is shown in red. And you can see they're showing us, in this example, it's about half as much. So our reactants are starting off with the same level of energy. We have to add this much energy, which, hey, that might be doable, to get our reaction going. And then our reaction will proceed and we get our products. So the enzyme doesn't change what reactants are involved, nor does it change what products are involved. It just changes what's our investment. And cells are good shoppers. We don't want to spend any more energy than we have to. When you're looking at your enzymes, you're going to be looking at the progress of the reaction in relation to the amount of product produced. So keep that in mind. This is how these things work. So the reactant that an enzyme acts on is called the enzyme's substrate. So we're talking about chemical reactions. It's a reactant, truly, but it's a special kind of reactant because it's something that binds to an enzyme, and that is the substrate. So if you want to take a look at your lab handouts, identify what the substrate is for the experiment you're going to be doing in lab this week. In the reaction, the substrate binds to the active site of the enzyme. So there's a special part of the protein called the active site. And that protein, remember, is our enzyme. Our substrate binds to our active site and is affected by the enzyme. We'll see this in picture form in a moment. The active site can lower the activation energy in multiple ways. So it's going to be specific for our enzyme and our substrates. One of the things that can happen is it can orient our substrates correctly. Another thing that can happen is it can take our substrate and encourage bonds to break or to form. And a third way that enzymes can work is they can provide a favorable microenvironment. So just the tiny environment of our active site can be a little bit different from the environment around the enzyme. And we learned about amino acids recently in class. We learned about some are charged, some are not charged, some are polar, some are non-polar, some are basic, some are acidic. And you can envision that the part of the protein that is in the active site has these amino acids which change the character of just that area. So in our active site of our protein catalyst, we could actually have a different pH, we could have some charge differences. Something is different about the little part of the enzyme that allows that chemical reaction to move forward more easily. So this is what it looks like. Here's our enzyme represented in this purple blob kind of thing. This is a very, very simple way of thinking about enzymes. So if this bothers you because you know it's harder than this, just relax and take Bio 201. Alright, so here's our enzyme. Here are our substrates or reactants in red and green. We're going to speed up our reaction by lowering the activation energy, convert those substrates to products, which are represented by the blue and yellow things, and release them into the environment. One of the important parts about this figure that's trying to show you is that it's a cycle. So our enzymes are going to go around and around, taking in substrates, kicking out products, taking in substrates, kicking out products. They are reusable. Unless they get damaged or conditions change, that enzyme will continue to crank out products. And then at the bottom here you see the example of sucrase that we watched in class. If you'd like to watch that again, absolutely go for it. So here's one of the things that you're going to be looking at in lab this week. It's the idea that each enzyme has an optimal temperature in which it can function, and it makes a lot of sense. So you're going to be looking at catalase enzyme at different temperatures. Each group will look at a different temperature. You'll compile your data, and then you'll share your data and analyze it. If you look on the left, you see an optimal temperature for a human enzyme. So we've got temperature along our bottom axis. We've got rate of reaction along our y-axis. When you graph your data, you're going to have temperature down here, and you're going to have mils of O2 produced per minute, or rate of reaction and mils of O2 produced per minute over here. So this is what we're looking at. If you look at this enzyme, this is a typical human enzyme, and its optimal temperature is about 37 degrees, which happens to be body temperature. I gave you there a second for those of you who just wanted to shout it out. So it makes sense that this human enzyme functions best at human body temperature. Now if you look at the red one on the right, this is temperature optimal for a thermophilic bacterial enzyme. The optimal temperature here is about 78 degrees, 77, 78. And if you look at the overlap, so we want to think about is, would this enzyme work in the human body? If you look at it, it doesn't even function at all over here at 37. So that's really important to think about too. When you look at maybe you go to GNC or the CVS pharmacy or something like that, and you look at enzyme supplements, think about the organisms from which those enzymes were isolated and whether or not that's going to work. There may be something interesting for you to discover. But each enzyme has an optimal temperature. Some of them actually have more than one. And you're going to be exploring the optimal temperature for potato catalase in lab. You're also going to be looking at the effects of acids and bases on enzymes in lab. So this is showing you each enzyme has an optimal pH in which it can function. And actually, again, some of them have more than one. So in the simple world, it's one. If you look at the curve on the left, this is a graph of optimal pH for pepsin, a stomach enzyme. And you can see the optimal pH is about 2. Well, we know our stomach is acidic, hence stomach acids. So it makes sense that a stomach enzyme would have an acidic optimal pH. Now on the right, we have the optimal pH for trypsin, an intestinal enzyme. Its optimal is about 8. So your intestines are actually much more basic than your stomach. But if we took this trypsin enzyme and we had it in the stomach, it probably wouldn't function very well. See, we don't even have any overlap there. So enzymes do have pH optimum as well. You will be exploring that a little bit in lab this week. So look over the enzyme section of the textbook, listen to this video again, watch this video again, read over your handout for the lab, and just make sure you're ready when you come in. And be sure to ask your instructor questions if you have any. Enjoy learning about enzymes.